Good afternoon. Today is Tuesday, December 8th, 2020, and we have reached the last lecture. This semester, every semester feels like this, but I think this semester in particular, you get that dichotomy where it feels like it is raced by so quickly, but at the same time, it feels like the class has been going on forever. It's such a unique experience that I think you get in a biology class where you get those two strong emotions at exactly the same time. And of course, this year it's been, truly unique in the way that we have handled this this semester, doing this all online and everything this way. You'll get to hopefully many years from now, we'll get to look back and laugh and look at the bright side. You will one day be able to say that you got to take anatomy and physiology from internet star and social influencer, Dr. Daniel Slutsky. So it's going to be, it's a feather in your cap, certainly. And in fact, for everybody who successfully completes the class with a passing grade, you'll actually get a free 6-month subscription to my OnlyFans site. So you've got that going for you as well. Just use the promotion code Syncytio Trophoblast. All right. Let's go ahead and start our game plan. The good news is we've covered most of the material we need to. We have to talk a little bit, touch on development. As I mentioned, we are going to hardly do justice to this because literally there are semester-long classes that are studying this material. But we're going to focus on some of the key highlights of it, the beginning and the end processes pretty much. And then hopefully there'll be some time at the end if there's any questions about the exams and things to go over, because that's all we have left after today. Thursday, you have your lab and lecture exam. You've already taken 4 of those. You know exactly what to expect from that. And then the following Tuesday, you have your final exam. Again, it is a cumulative exam, 100 multiple choice questions, 100 minutes to complete it, must be completed during class time. And again, covers everything that we've covered in this class. It's completely cumulative, but at the same time, remember, they're all multiple choice questions. So you don't have to pull the information out of the ether. You just have to recognize the correct answer. And that is a very, very different technique. So hopefully read the questions carefully so you can answer them correctly and you'll do well on that. Remember, that final exam counts as a grade on its own, but it also can be used to replace your lowest lecture score. All right, and then Wednesday the 16th, you get to sleep or drink, whatever tickles your fancy. All right, questions on any of that? All right. So we left off last time and we were talking about, oh, excellent. We were talking about development, and we talked about some of the vocabulary. And as we said, we were going to focus on this process of development from fertilization to implantation. Oops, where is my annotations? There it is. All right, so as we know, 2 haploid nuclei are going to merge together to form one diploid cell. And that process, that cell that is formed from that, as we know, is called a zygote. We have our spermatozoon. We have our secondary oocyte, frozen in metaphase 2. And remember, when that sperm is able to penetrate the secondary oocyte, it completes meiosis 2, becoming that mature ovum. So we have the mature ovum with a haploid nucleus. We have the mature spermatozoon with its haploid nucleus coming together to form a single cell with 46 chromosomes that we call a zygote. As we talked about, the female reproductive tract from the vaginal canal to the ampulla of the uterine tube is only about 5 inches. So it is a relatively short journey that sperm has to take. Some of them can complete it in as little as a few minutes. Others may take hours to be able to complete that journey. But as we also talked about, it is a very treacherous journey with an incredibly high attrition rate. Remember, as we talked about, in the average ejaculate, there are 500 million sperm that are released into the vaginal canal. So 500 million sperm in an average ejaculate. Of those, only about 200 million actually make it up to the top of the vagina. From those, only about 2 million reach the cervix. Of those, only about 10,000 reach the uterine tube. And of those, only a couple hundred actually reach that secondary oocyte. So remember, we talked about a male who has an ejaculate as little as 150 million sperm per ejaculate is essentially infertile. And now you see why, because with the attrition, only a small handful, 4 or 5, would be able to reach that secondary oocyte. And as we've talked about, that secondary oocyte has defenses. You need hundreds of sperm to reach it so that those defenses can be penetrated so that fertilization can take place. So it's one of the reasons why males produce such a massive amount of spermatozoa, is because a massive amount is needed for successful fertilization. This obviously is an illustration, but it does give you a sense of the scale of the size of that sperm to the size of the secondary oocyte. So again, as we've talked about, that spermatozoa are just chromosomal missiles contained in a little haploid nucleus that it is presenting to the egg. Whereas that mature ovum needs to have not just half of the chromosomes, but all of the cytoplasm, all of the rough endoplasmic reticulum, all of the mitochondria, all of the Golgi apparatus, so that it can be a viable cell. And that's, again, why we get that uneven division in females. We still need to divide those 2 times or 4 times to get 4 cells, 3 or 4 cells, depending on what that primary polar body does, that first polar body does, but it divides unevenly. So basically we get polar bodies that just have the chromosomes so that this cell has the right number of chromosomes, but it keeps all of the cytoplasm. All right, so again, let's talk about what has to happen before fertilization. Obviously, we have to first have ovulation. That secondary oocyte, frozen in metaphase 2, has to be released from the uterine tube. As it is released from the ovary, and hopefully gets into the uterine tube. Again, as we talked about, not all of them make it that way. So remember, great question. Remember, with the first division, 1 becomes 2 cells. One of those cells is our first or primary polar body, and the other is our secondary oocyte. If fertilized with the sperm, that secondary oocyte divides. And when it divides, we have the mature, not sure what that was. We have that mature ovum and the second polar body. So we could have 2. But remember also, it is quite possible for this first polar body to divide. And if this first polar body divides, then we would have 3 polar bodies and 1 ovum. So we can either have 3 or 2, depending on this. This stage, this second division, that's not even close to being right. Sometimes happens and sometimes doesn't happen. And quite frankly, we don't care one way or another. All of these polar bodies are going to just be fuel for the rapid division that needs to take place. After all, these cells, this one zygote needs to divide gajillions of times to make gajillions of cells. And so these resources, especially that DNA, those nucleotides, can be used for the rapid division that it needs. So whether we're breaking down 1 cell or whether we're breaking down 3 cells, or 2 cells or 3 cells, doesn't really matter. Here's the other thing that I don't think we necessarily emphasized at this point. Remember, all of this is taking place inside of that zona pellucidum. So when that cell divides, we have, let's not do that. We have our mature ovum and those potentially 3 polar bodies all still inside that glycogen-rich, candy-coated shell that is the zona pellucida. So all of this division is taking place inside of that shell. So again, these are just resources in there with what at this point would be a mature ovum with a sperm inside of it. And so those are resources that are still inside of the zona pellucida, inside that candy-coated shell. All right, so ovulation occurs. Our secondary oocyte enters into the uterine, and thanks to the cilia of those simple columnar epithelial cells, it gets moved about 1/3 of the length down the ampulla, down the uterine tube to the ampulla. Now, what we haven't talked about is there's 2 uterine tubes, and we talked about all this attrition that only 10,000 make the uterine tubes. Is it because half go to the left and half go to the right, and they just hope for the best? No. Is it possible that some will go the wrong direction? Yes. However, that secondary oocyte is releasing a chemical attractant to make it much more likely that the sperm will go into the correct uterine tube and much less likely that they will go into the wrong one. So I think it's something like an 80-20 split or something like that. Again, some of them are not as sensitive to the chemicals, some may not come in contact, and so just random movement may get them to go the wrong way. But the majority are going to go to the correct uterine tube because of that attractant that is being released by the secondary oocyte. Again, the sperm must swim through the uterus, through the uterine tube. But remember, we also talked about, thanks to the prostaglandins, and if the female is lucky, thanks to orgasm, we will get a reverse peristalsis of the vaginal canal and the uterus. And those uterine contractions, those vaginal contractions, can help to draw the sperm up, increasing the likelihood of them getting there, helping to facilitate that swimming process. However, and again, it's like I've talked about before, in all the cartoons and all the movies, you all see all the sperm swimming and racing, and it's always like the first one there that wins, but that is not the case. As we've talked about, they have those defenses they have to work their way through. But it also takes the sperm some time before they're capable of defeating those defenses, of doing the work necessary to get in. This process of breaking down the defenses, of releasing our digestive enzymes, of changing the swimming motion, is a process we call capacitation. So even if we have this egg and this sperm in a petri dish and we just plop it on top of it, it's not going to be able to instantly fertilize that egg. It can take up to 8 to 10 hours for the sperm to be capacitated, right? Which means that it is capable of fertilizing, beating those defenses, and hopefully penetrating those defenses to fertilize the egg. This capacitation process is twofold. One, it makes the acrosomal cap fragile so that it ruptures, releasing those digestive enzymes. Those digestive enzymes are going to break down that glycogen-rich barrier that is the zona pellucida. And it changes its swimming motion of its tail to kind of dig the head of the sperm against the surface of the zona pellucida, trying to work its way through, to basically dig those enzymes into it to break it down and try to tunnel through that zona pellucida to get inside. Not surprisingly, this takes a tremendous amount of energy, a tremendous amount of resources, and the sperm is only able to do it for a few minutes, at which case it then exhausts itself and basically becomes non-functional. But then the next one's right behind it, and the next one's right behind it, and the next one's right behind it. And so it takes hundreds of sperm digging away at that zona pellucida to finally penetrate through where one is finally able to get in and make contact with the actual plasma membrane of the secondary oocyte. And that's the goal. I've described it in words. Here we see a pretty picture of how this process works. Again, as we see, our secondary oocyte has already undergone meiosis. Now notice this one's been fertilized, so notice it's completing meiosis 2, but it had already completed meiosis 1, so we have our first polar body in there. But what I really want to talk about are these defenses. Again, as we talked about, we have those granulosa cells that form the corona radiata on its outer surface, and that glycogen-rich hard sugar shell known as the zona pellucidum. Now, on the surface of the zona pellucidum is a special receptor called ZP3. This ZP3 receptor is actually what causes the sperm to release its digestive enzymes. Remember, we talked about how the acrosomal cap becomes fragile. It is contact with that zona pellucida receptor, that ZP3 receptor, that causes it to release it and allows it to dig that in to try to beat away at the zona pellucida, and that's what we call the acrosomal reaction. So again, our sperm binds to the ZP3 receptor, our acrosomal cap opens up, releases those enzymes, and our sperm starts to grind away at the surface of that zona pellucida, trying to penetrate its way through. Like I said, this process lasts for a few minutes, and then basically it exhausts itself and becomes non-functional. In this fashion, it takes hundreds of sperm to get their way in. Yep, ZP3 receptors are on the zona pellucidum. So first they have to dig through the corona radiata, squeeze through those cells to get to the ZP3 to release that digestive enzyme to break away at that glycogen-rich shell. And that's why I said, there's no advantage to being the first one there, right? The first 200 that come are the big workers. They got up early, they got there, they did their job. And then it's that slow, lazy one, the one that stopped for ice cream on the way, that comes strolling up several hours later after all the other ones have done all the hard work, that get to slip inside and actually fertilize the egg. America. All right. Now, the goal here is to get a sperm inside to fertilize that egg. But the goal is just to get one sperm inside to fertilize that egg. All right. So when this occurs, when that opening finally occurs, so let's draw this. So here is our zona pellucidum. Inside of the zona pellucidum is the plasma membrane of the secondary oocyte. And of course, on the zona pellucidum, oops, let's make them blue. Are the ZP3 receptors. So our sperm have been chewing away at the zona pellucida, binding to those ZP3 receptors, releasing their digestive enzymes, and working their way through the zona pellucida. So our goal is to get that first sperm to come in contact with the plasma membrane of our secondary oocyte. Now, there are special receptors. Whoops. Hold on. On the surface of the plasma membrane. And again, in another misnomer, I guess, for lack of a better term, the sperm doesn't actually penetrate the egg. What the sperm actually does is come in contact with that receptor. And when it comes in contact with the receptor, then the egg endocytosis there you go, the head of the sperm. So the sperm doesn't even penetrate it. It's actually the egg that has the control and the egg that brings it in. And that is how fertilization is going to occur, right? But again, remember the key is we want one and only one sperm to fertilize the egg. This goal of having one and only one is the goal of maintaining Syngamy, only allowing one sperm to fertilize the one egg. Now, the way this occurs starts with what happens when that head of the sperm binds to the receptor. When the head of the sperm binds to the receptor, two events occur to maintain this Syngamy. This is the fast block and the slow block. The fast block, basically three things occur. The fast block is that we obviously get the endocytosis, the head of the sperm. But two other things occur as well. The first is that the cell undergoes a massive depolarization. This depolarization helps to inactivate the receptors on the plasma membrane, so that even if a second sperm somewhere else comes in contact with it, it won't be drawn inside. The third thing that happens is that depolarization leads to the massive release of calcium inside the cell. And as we know, calcium makes cells do wonky things. And in this case, the wonky thing it is going to do is that slow block. Calcium makes cells do wonky thing. One of the wonky things that it's going to cause the cell to do is release a chemical that inactivates the ZP3 receptors. Basically, if we can inactivate the ZP3 receptors, no more sperm are going to be able to bind to it. No more sperm are going to release their digestive enzymes. No more sperm are going to try to dig their way in. Next, not only does it stop the release of the acrosomal enzymes, but it also thickens and strengthens the zona pellucidum. This fertilized egg has to actually live in here for a period of time. So it is actually going to thicken and strengthen the zona pellucidum. Again, making it harder for any other sperm to find their way in. And then, of course, we can't forget the last thing that calcium does. It triggers the completion of meiosis 2 so that we get our mature ovum and our second polar body. So in this fashion, not only do we get the fertilization of the egg, but we help to maintain the fact that only one sperm and not more than one gets in. If more than one gets in, yeah, the calcium is what is responsible for the slow block. That is correct. If a second sperm gets in, a condition we call polygamy, is that going to be a viable fertilized zygote? No, and that basically won't be able to divide, won't be able to come mature to term, and won't be a baby. Exactly. All right, I've done a poor job of showing this with my pictures, but let's go back to the lecture and talk about this. So again, we need that membrane to fuse. So the sperm binds, the first sperm binds the plasma membrane, and we get that fast block where the membrane fuses to block multiple sperm from coming in, that condition we call polyspermy. That fast block occurs 1 to 5 seconds after contact. Our oocyte membrane depolarizes rapidly, releasing a massive amount of calcium from the endoplasmic reticulum. And again, don't forget, this is also where we get the endocytosis of the head of the sperm. Not the whole sperm gets to come in, just the head. The tail basically is broken off and left outside. That calcium triggers our slow block, or what is called the cortical reaction. It releases a special protein called the zonal inhibiting protein. And that zonal inhibiting protein inactivates the ZP3 receptors so that no more sperm can bind to them. And as I said, it thickens and hardens the zona pellucida, so any that are digging away at it can't get in. Lastly, it triggers the completion of meiosis 2. I did an okay job of drawing this, but I do actually like the picture from your textbook. Your textbook does a really nice job of showing this. And here we see that process. Notice here we see our zona pellucidum with the corona radiata. Our sperm with their acrosomal caps intact are digging their way in, and they make contact with a ZP3 receptor. When they make contact with that ZP3 receptor, it ruptures the acrosomal cap, releasing those digestive enzymes and causes the sperm to dig away, trying to get its way in. Many sperm are required to break through until finally one sperm is able to get in and come in contact with a receptor on the plasma membrane of the egg. When it does that, that triggers a fast block, a massive depolarization, which inactivates these receptors, a massive release of calcium from the endoplasmic reticulum inside the cell, and the endocytosis of the head, the nuclear component. And we have basically our haploid nuclei located here inside of the oocyte. That massive release of calcium triggers our slow block or cortical reaction, where we release that zonal inhibiting protein that inactivates the ZP3 receptors and hardens and thickens the zonal pellucidum, and it triggers the completion of meiosis II. So the question I was just asked is, do some run out of, I'm not sure but basically run out of energy, I guess, just as they finish digging through and just slip away? My guess is that once they penetrate through OATP, once they penetrate through, they probably are able to, because again, the plasma membrane is right up against it. So when you bust through, my guess is that you come in contact with the plasma membrane, and the one that finally ultimately breaks through probably gets it. But is there one that gets probably all the way there and isn't able to, can see it through the thin membrane but doesn't get all the way through? Probably. Right? Again, you got hundreds of these working at this in different locations trying to all fight their way in. But my guess is the one that finally bursts through probably has enough, if nothing else, momentum to bring it through and get it in contact with the plasma membrane. All right, enough of the pretty pictures. Let's actually look at some light microscopy. When that egg is fertilized, when it completes meiosis II, what we end up with, as we said, is that zygote, where basically we have a single cell, that mature ovum, that has two what we call pronuclei. Right? This one here is the nucleus from the head of the sperm. This one here is the nucleus from completion of meiosis II. And they are still bound in their plasma membranes. So what basically starts to happen is this cell now sees a dramatic increase in metabolism. And it starts mitosis. Mitosis is, of course, the process of dividing one cell into two identical cells. And of course, in prophase, as we know, those nuclear envelopes start to break down. And as those nuclear envelopes start to break down and our individual chromosomes start to form, so that loose chromatin packs up into those tightly packed chromosomes, at this point, all the chromosomes are together inside of this one cell. We now consider this cell to be diploid, having the 46 chromosomes, and it is now a zygote. And the job of the zygote? Division. This zygote is going to undergo very, very rapid division. So again, here we kind of see this process. Notice here we have our secondary oocyte that was frozen in metaphase II. The sperm is endocytosed. Anaphase, meiosis II is completed. Notice we end up with 3 polar bodies and our ovum with that pronuclei of the male. We get our 2 spindles, centrosomes that form, the breakdown of the nuclear envelopes, chromosomes come together, and at this point, we now have a zygote that is in prophase of mitosis, starting its division. Now, one zygote, one baby. But as we know, is it possible to have more than one baby? at the same time? Absolutely. And when we have those types of twins, basically they come in two flavors, right? Or triplets or quintuplets or whatever it is. Monozygotic and dizygotic. What's the difference? True, we do have, Kate and whatever plus 8, where they had 8, well, actually they had two twins and then 6 at the same time. But typically you get that with in vitro fertilization. You get a doctor who is willing to put massive amounts of fertilized embryos into the mom in hopes that many of them take. Normally when you do, well, okay, let me say that. It is not uncommon with in vitro fertilization for multiple embryos to be placed into the female in the hopes that at least one of them will take. However, when a doctor puts 12 of them in there, the likelihood that more than one will successfully implant is highly likely. And that dramatically puts the female and the babies in much more stress and much more danger from that process, and is incredibly, while not technically illegal, although it is illegal in some states, while it is technically not illegal, you could argue it is unethical and certainly not wise. But going back to the idea of just twins, you have the right idea. The difference between a monozygotic and dizygotic is whether there's one or two zygotes, or really one or two secondary oocytes. In the case of a monozygotic twin, what happens is you get one egg and one sperm that come together to form one zygote. As we'll see, that zygote is going to divide many times to form a ball of cells. And sometimes during that process, that ball of cells will separate into two separate balls of cells. And those two separate balls of cells divide to become two very separate different people, but they have the same genetic information. And so, as someone pointed out, we refer to them also as identical twins.
What can cause this?
Great question. From what we can gather, it's fairly spontaneous, right? Dizygotic twins are actually quite, are strongly influenced by genetics. So if you have dizygotic twins in your family, you typically have many of them. That happens a lot. It's common in, like I said, there's a strong genetic predisposition for that. And basically what happens is the female, and it's on the female side, the female is more likely to mature and ovulate multiple eggs. So when that occurs, you can have multiple kids because each egg gets fertilized by a different sperm. And so then you can end up with a Brandon and a Brenda, right? In fact, there have been some cases where you have dizygotic twins, two zygotes, two eggs that are released that are fertilized by two different males. So you have twins that are born that are actually half siblings, same mom, different dads. But the division of the cells seems to be more of a random event. It doesn't seem to run in families the same way. So when you get identical twins, it tends to happen more randomly. And of course, in some cases, that division occurs very late and you don't get a complete division. So what ends up happening is you have two people who share a liver and are still connected together. What do we call that condition?
Conjoined?
Conjoined, exactly. Conjoined twins can happen when you don't get a complete division of those of that zygote. All right. Questions on that? All right, let's talk about these divisions. One of the things that happens is this cell becomes highly metabolic and divides rapidly. And one of the keys to dividing rapidly, normally when we think of mitosis, if you think way back to 430, one of the ways we could tell a cell was in interphase or even in prophase is that it had gotten really large. Because typically before one cell divides to become two cells, it gets really big, so it has enough stuff to become two cells. Here, what's happening, yes, here what is happening is that The cell is dividing so rapidly, there's really no time for it to grow. So one cell becomes two cells that are the same size as the original. Two cells become four cells that combined are the same size as the original. So with each division, the number of cells go up and the size of the cells go down. Yes, a chicken egg is a single cell, one cell. In fact, if you've ever seen an an ostrich egg is a single cell. In fact, I believe it's the largest single cell on the planet. So with these rapid divisions, we produce more cells called blastomeres, and you get more of these cells, but the cells get smaller. And that way, notice it all stays inside of that zona pellucidum. So after about 30 hours, this rapid division is what we call cleavage. Our one zygote divides to become two blastomeres. By day 2, our two blastomeres have divided to become 4. By day 3, we get a cluster of about 16. And it starts to form a solid mass of cells. And this solid mass of cells is what is known as a morula.
May I ask you?
Yes.
So those cells can be considered as stem cells?
Yeah, yes. At this point, they are essentially omnipotent stem cells. Completely. And think about it this way. Let's go even more dramatic. Let's say, for argument's sake, I took this cell, this cluster of cells at day 3, day 4, that had somewhere around 20 cells in it. If I were to say, for instance, take this cell, pluck it out, and switch places with it with this cell here, would this person suddenly have a foot where their hand is and a hand where their foot would be because I changed the position of those two cells?
Not yet.
No, not yet at this point. At this point, these are completely undifferentiated cells. They're completely undifferentiated, omnipotent stem cells, absolutely.
Okay, thank you.
And by about day 4, we have this massive, dense cluster of cells of about 100 cells. And again, it is a uniform mass. At this point, at about day 4 or around day 5, it actually enters into the uterine cavity. Remember, as we talked about, it takes about 3 or 4 days to get the egg down from the uterine tube into the uterus. But around day 5, something different starts to happen. As it enters into our uterus with that thick, rich, glycogen-rich uterine milk, it continues to divide, but it also starts to differentiate. Notice from the outer surface, it just looks like a solid mass of cells, just like we saw before. But if we take a cross section through it, what we can see is that there are basically 3 distinct regions. The first of these regions, and let's highlight them in color. The first of these is this dense pack of cells located here on the inside, known as the inner cell mass. However, there is a thick shell of cells on the outer surface, and that shell of cells on the outer surface is what is known as the trophoblasts. Great question. Hold on, let me finish this real fast. And then what we have in between is a fluid-filled space. So notice in here, we have this fluid-filled space known as the blastocyst cavity. Let's take a look at a different picture that shows this. Oh, I thought I had a better picture for this. Well, I guess this one over here is not bad. Trophoblast, inner cell mass, and this and cavity. We see that there. We see it here pretty nicely, but we'll come back to this one here. All right, the question is, at this point, they're still completely identical and omnipotent. They are still omnipotent, but at this point, they are starting to differentiate. At this point, the cells have different goals, different functions from them. And so at this point, this cell out here is not going to necessarily become the same thing that this cell here is. And so at this point, we're starting to get some differentiation will occur. Again, if we were to take two of these inner cell masses and switch them around, again, you're not going to get a hand where the foot is and the foot where the hand is, but you are starting to see some differentiation, especially later on in this stage. Uterine milk is what is being produced by, remember, in our uterus, we talked about during that secretory phase, the post-ovulatory phase, we have a massive glandularization, releasing a massive amount of glycogen-rich secretions. And those glycogen-rich secretions from the those uterine glands is called uterine milk. It's not glue. It's not what's going to attach it to the wall. We'll see that in just a second, how that occurs. But that's not what attaches it to the wall. It's just sustaining it. And remember, at this point, it's all still occurring inside of our zona pellucidum until ultimately it hatches out. And about day 7 is when it hatches out of that, about 5 to 7, it hatches out of the zona pellucidum. So we've looked at these with illustrations, but let's actually, I did a quick YouTube search. And let's look at some of the examples of this that we can see. So which one do I want? I want this one first.
One day after fertilization, the embryos are observed under the microscope. If fertilization has occurred, a single cell is seen containing 2 nuclei.
So again, as you can see, this is where we have, there's my annotation, the 2 pronuclei, one from the egg, one from the sperm, that have not fully come together yet.
Male and female. Two days after puncture, the embryo divides itself and contains 2 cells with 1 nucleus per cell.
And again, we call
which
those,
in turn starts to divide into an embryo of 4 cells if its development is optimal.
and we call those blastomeres.
3 days following puncture, the embryo starts to divide and consists of 8 cells if its development is optimal. On day 4, the embryo divides a number of times, becoming a morula. This is when it begins to become compact. On day 5, the embryo contains more than 100 cells. In the center, there is a fluid-filled cavity. Finally, the blastocyst will shed the membrane that surrounds it. This is called hatching.
So here we saw with the nice pictures, but what I also want to show you is an actual time lapse of this, where we can actually see these events. It goes fast and doesn't show.
What's the difference between an embryo and a blastocyst? If you want to find out more, please watch this video. I'm Dr. Malpani. I'm an IVF specialist, and one of my goals is to educate patients about IVF so they have realistic expectations and don't get taken for a ride by doctors. Now, fortunately, because IVF has been around for so many years, patients are slightly better informed about what's involved in an IVF lab and a clinic. So at least they're familiar with words like embryos and blastocysts. But the trouble is they often don't know enough. And the worst thing is, a lot of times that doctors will lie to them or try to bamboozle them by using jargon which is completely inaccurate. Partly because the doctor doesn't know any better, because remember, he's not an
All right. I was wondering where the sound was coming from. I was very confused. Let's try this again. All right, so here we have our single cell. Notice we even have the polar body out here. And this single zygote divides to become 2 blastomeres. The 2 divide to become 4. And the division continues. The 4 become 8. All do it in the first 3 days. Then from day 3 to day 6, that division continues. 8 becomes 16. And again, at this point, it is a solid mass of cells known as a morula. They condense down, continuing to divide till hundreds of cells are present. And when about 100 cells are present is when we start to see the differentiation. Notice now the cells are pushing apart from each other. Oops, too fast. Come on, hold on. Let's go back a little bit. So we are starting to get the formation of the inner cell mass. We can start to see the void that is the blastocyst cavity. The trophoblast is starting to form on the outside. And notice now, finally, it's starting to penetrate through and break out of that zona pellucida. And that process we call hatching. And there you go. They didn't show the full hatching, but again, I just did a quick search for this stuff this morning, and that was what I found. All right. Questions on that? Excellent. I think as far as I can tell, everybody completely understands this because no one is saying anything. Awesome. All right, excellent. So at this point, we have now formed our blastocyst. And it is about day 6 or 7, 5, it basically hatches and it hangs out for another day or 2 until final implantation takes place. Now, before we discuss implantation, this is a good stopping point. It's a little bit early for our first break, but I think this is a good natural stopping point before we get into some more vocabulary. So let's go ahead and take our first break here. We'll take a 15-minute break. So we'll say 1:10, come back at 1:10, and at 1:10, we'll pick up from here. Any questions? All right, see you guys in 15 minutes. All righty. So, any questions before we dive back in? All right, we left off last time, and as we mentioned, at around day 5, day 6, the blastocyst hatches from the zona pellucidum, and it continues to kind of float around inside the uterine milk, released into the uterus for another couple of days. Then typically around day 7 or 8, a week after fertilization has taken place, it is going to attach to the endometrium. Now notice the first thing that has to occur for this to take place is our blastocysts have to rotate. Remember, now we're starting to see some differentiation. And so what's going to happen is that the inner cell mass is going to turn so that it faces the uterine wall. And when that occurs, it then makes contact with the uterine wall, with that functional layer, that big, thickened, vascularized, glandularized functional layer. Now, this typically occurs within the body or up at the fundus at the top. Thankfully, usually it doesn't occur inferiorly closer to the cervix. And as you look at the picture here of the uterus, you could see why that would be a problem. Where it attaches is where the placenta is going to form. And if it attaches too inferiorly, what can actually happen is the placenta can actually form and develop over the cervical canal. And if it forms over the cervical canal, is this baby going to be able to be expulsed in a normal type of birth? No. In fact, typically it has to be delivered relatively early. Because if you wait to the point where the cervix dilates, that dilation of the cervix can actually cause a tearing of the placenta, and the placenta is incredibly well vascularized, and that tearing of the placenta can be incredibly dangerous for baby and for mom. And then clearly you're not going to be able to push the baby out if the placenta is in the way. So it's going to be a much more challenging birth if that placenta forms over the cervix as opposed to forming up here in the fundus or up here in the body. Now, when it attaches to the functional layer, we see an elaborate transformation take place in the trophoblast. Actually, there's two elaborate transformations that are taking place. One that is taking place in the inner cell mass, and we'll talk about that in a second, but also one that takes place in the trophoblast. There's a question. It's not that it's too small of an area, it's that it would block the opening, and then also the dilation of the cervix could cause a potential tearing of the placenta, as we'll see, which is a very vascular structure. So it could be incredibly dangerous to mom should the placenta tear in that fashion. So it isn't that it's too small of an area for placenta to form, it's that that's the exit. And if you cover up the exit, that's going to cause problems. All righty. So as I mentioned, we can see here basically that our trophoblast, that outer layer of cells, undergoes a bit of a transformation and starts to form two distinct layers. The first, and again, this is one of those alphabet soup words you can be pretty much guaranteed you're going to have to spell, is the syncytiotrophoblast. The syncytiotrophoblast would be the part that's here represented in the light yellow. Basically, what happens is these cells enlarge, the trophoblast cells enlarge and expand. And they secrete digestive enzymes. And these digestive enzymes start to digest away the functional layer of the uterine wall. This allows the embryo to start to, or literally blastocyst at this point, to embed itself within the wall, but also hollowing out the functional layer is going to ultimately leads to the development of the placenta as well. However, notice we still need to maintain the integrity of the blastocyst cavity where the inner cells are located. And so we still maintain a layer of cells forming that boundary, and that is called the cytotrophoblast. So this maintains the original shape of the embryo while the other cells expand and grow inward, burrowing into the functional layer. This burrowing continues until the blastocyst is completely embedded within the functional layer, completely contained inside. So notice our syncytiotrophoblast continues to expand and it continues to grow until it is completely embedded, so that the functional layer now closes over the top. It is completely contained within the uterine wall. Now remember, it's able to do this in the functional layer because remember, our trophoblast cells are producing what produce that human chorionic gonadotropin. And it says here it maintains the uterus, but remember what it really does. What it really does is maintains the corpus luteum. And the corpus luteum continues to produce estrogens and progestins, and those estrogens and progestins maintain the uterus. Sometimes when individuals have difficulty having offspring, one of the problems they can have is not so much a problem with the fertilization of the egg, but actually this process of implantation. What happens for these individuals is fertilization isn't an issue, but for whatever reason, the cells don't produce enough human chorionic gonadotropin, or as a result of that, the ovary is not as sensitive to the human chorionic gonadotropin, so it's not able to maintain that large corpus luteum. And what happens is progesterone levels will drop. When this occurs, when people have difficulty getting pregnant with this type of an issue, then often what happens after ovulation, after copulation takes place, they will then take progesterone supplements to help to maintain the progesterone levels, to help to maintain the corpus luteum so that implantation can take place. So the syncytial trophoblast is going to help to form the placenta. And that placenta is going to be what produces the hormones, the glandular structure that produces the hormones that replaces the corpus luteum. So it isn't that it's forming a new corpus luteum. First, it is producing the hormone, human chorionic gonadotropin, that maintains the corpus luteum. And then it forms the placenta. And then the placenta really produces and releases the hormones. So it becomes responsible for the hormones instead of the corpus luteum. Okay. So yeah, so remember, the difference between the embryonic stage and the fetal stage is really who's producing the hormones. During those first 9 weeks, it is the corpus luteum and the ovaries that are producing the estrogens and the progestins. After week 9, the placenta has formed and it is producing the estrogen and progestins. So yes, after the placenta forms, the corpus luteum is no longer maintained. And no, and yes, you are correct. That's what human chorionic gonadotropin does. Luteinizing hormone levels still drop, and normally that would cause the corpus luteum to shrink. But instead, human chorionic gonadotropin maintains the corpus luteum. So even though luteinizing hormone levels drop, human chorionic gonadotropin maintains the corpus luteum. So yeah, so the corpus luteum doesn't have to rely on luteinizing hormone anymore. Instead, the human chorionic gonadotropin maintains it. All right. All right, so as we mentioned, the syncytial trophoblast penetrates down, digs the hole so that the egg can burrow inside. But notice the other thing that it does is it expands within this space, forming these large openings, forming these large expansions. And notice what starts to happen is the cytotrophoblast starts to expand into those spaces. And as the cytotrophoblast expands into these spaces, it forms structures that are called the chorionic villi. These chorionic villi, this is so large. These chorionic villi are going to contain large capillaries. Notice these capillaries are surrounded by large blood-filled spaces. The other thing that the syncytial trophoblast does is it forms these large spaces in the functional layer. And as we know, the functional layer is very well vascularized, so there are going to be many blood vessels feeding into and out of this space. Many arteries and veins feeding into and out of this space. And so basically what ends up happening is it becomes this big, huge blood-filled space. And this blood-filled space basically becomes the intervillous sinus, or the intervillous space. This is the beginning of the formation of the placenta. Notice in this location, what happens is the blood, and I think I actually have one more picture, so let's switch to the next picture. Nope. Here we go. Perfect. Here, this structure basically becomes the placenta. Notice here we have those chorionic villi containing the capillaries. We have all the maternal arteries feeding blood into and veins drawing blood out of this fluid-filled space, blood-filled space. And again, this blood-filled space is known as that intervillous sinus. So let's write that. I'm not seeing anything that points at it. If it is, I'm not catching it, but that's all right. And notice in this location, the blood supplies from mom and baby come close, but do not mix. All right, that's the real key to this. Mom's blood is feeding into this vast pool that is going to contain all sorts of oxygen, all sorts of nutrients, all sorts of chemical signals. And basically baby's blood's here and going to come here. And this is where the exchange is going to take place. The blood supplies are in close proximity to each other, but the blood supplies don't mix. Now notice we don't just want capillaries from mom coming together with capillaries from baby, because as we know, the conditions of our blood can change dynamically, right? Despite what many people think, are pregnant women truly eating all the time? No. And hormone levels can be changing all the time. And we don't want those minor fluctuations that are happening moment to moment to have an effect and to have an influence on the baby. So by having this large pool in this intervillous sinus, it accumulates hormones, it accumulates nutrients, it accumulates oxygen, so that all those things can be given to baby. And this is how the baby gets rid of its carbon dioxide, rid of its waste, rid of its other materials that way. It can give stuff to mom, and then this blood gets constantly circulated. Yeah, absolutely. We want to keep baby cells and mom cells separate from each other, because baby cells, because half of their DNA comes from dad, is going to be making proteins that mom's body is not going to recognize. If you remember way back at the beginning of this class, when we talked about blood types, we talked about the Rh factor. If mom is Rh negative, but baby gets the Rh factor from dad, her red blood cells will have that Rh protein on their surface. And mom's blood has the Rh antibodies. And that's one that can cross this placental barrier and attack baby's blood. So that'd be a bad thing. Most antibodies, most things can't cross, but that Rh factor is one that can. So the blood supplies don't mix. They come in close contact, but they don't actually mix. And it is that syncytiotrophoblast that not only forms the spaces, right? So not only does it implant the egg into the functional wall, but basically forms that intervillous sinus that is going to be mom's part of the placenta. And then, as we said, it is the cytotrophoblast that expands to form the villi. All right, we jumped past a slide here, so we'll come back to that one in a second. And so that is what is happening with the trophoblast. But notice also, as we look at these pictures, there's a whole lot going on with the inner cell mass as well. So we can briefly talk about that, and it really is truly going to be briefly. That inner cell mass basically differentiates and forms three main embryonic cell layers. The first is a cell layer known as the endoderm. This is what forms the epithelium and the epithelium of the gastrointestinal tract and the respiratory tract. Our mesoderm, which forms the muscle cells, the bone, and our other connective tissues. And the ectoderm. The ectoderm is what forms the skin and the nervous system. During this process, there is a process where the cells basically fold in on themselves, a process known as gastrulation, which is basically where the organ systems develop, and neurulation, which is the process of the development of the nervous system. And again, these are big, huge, elaborate processes that we could talk for years about, but we're not going to do that. So just know those terms, what they mean, know the 3 embryonic cell layers and what they basically become. And that's all you're going to need to know for this. Like I said, again, we are not doing any service to this stuff. This is something that can be covered in a whole semester, and we're doing it in, a couple hours. But we're getting the introduction to it. All right, excellent. As I mentioned, starting about week 10, that placenta forms. Again, the trophoblast and form the chorionic villi for the baby, mom's functional layer, and that intervillous spaces where we get the close proximity, but not the actual crossing of blood from one to the other. Again, we get oxygen, nutrients, wastes, the hormones. All those things are stored in that blood and provided for a baby. And again, it's just forming the placental barrier so the two bloods don't touch to provide that protection for the baby. Upon birth, the placenta is also expulsed. Not just the baby comes out, but the placenta has to come out as well. It needs to be inspected to make sure it's intact, because again, it is a very vascularized structure. So if left inside, there can be significant rapid blood loss for mom. But also, even if it's not a blood issue, remember the placenta is the major hormonal structure. If a portion of the placenta stays behind, the hormones that are produced by the placenta can affect mom's ability to be able to recover from the pregnancy and to also potentially start to produce milk. So it can have all sorts of issues if it is still in there producing all of its hormones. Here we see a picture of the placenta and then the other structure that connects that placenta to the baby, the umbilical cord. Umbilical cord is about 24 inches long. Inside it, it has 2 arteries and 1 vein. And again, normally we think of especially systemic arteries as being oxygen rich and veins being oxygen poor. Is that going to be the case here? No, exactly. The mom is providing the oxygen to baby, so it is going to be the umbilical vein that carries that oxygen-rich blood back to the body, and the arteries coming out carry the waste. Now, one of the interesting things about this, I see your question, Jessica, and I'll answer it in just one second. Let me finish this first. Nutrients, obviously, again, come in the vein, and wastes go out the arteries. Notice they come together to form the umbilical cord, which attaches to the umbilicus. But notice where the arteries and the veins come from and go to are very different. Notice the 2 arteries actually come off of the internal iliac arteries, bringing the waste, bringing the carbon dioxide to the placenta. Whereas the vein brings the oxygen and brings the nutrients. And of course, as we know, it brings it to the liver to be processed. And of course, as we talked about, when we bite off the umbilical cord and it dries up to be a belly button, we also know that that umbilical vein also shrivels up to be that ligamentum teres that we talked about in the liver. All right, so there's a question, is there any scientific evidence that eating the placenta is beneficial to the mom? So here is what I would say for that. Certainly most animals eat the placenta. The placenta is a structure, especially if they're out in the wild, it is a structure that is very nutrient dense. And mom having given birth and having to nurture the baby, having those nutrients are something that is definitely important. However, as we also know, not only is it a very nutrient dense structure, but there's also a tremendous amount of hormones. And so there is some evidence that shows that ingesting the placenta can assist in lessening symptoms, excuse me, symptoms from things like postpartum depression and things along those lines. It can help to stabilize hormone levels because you've got these massive fluctuations in hormones taking place during pregnancy and even more fluctuations that are taking place at birth. And recovery for that for some women can be very challenging. And so there is some evidence that shows that the ingestion of the placenta can potentially be beneficial. There are also certain cultures that ingest placenta as part of their cultural beliefs. Others will use it as fertilizer, basically to plant with a tree, so that you have a tree that's born at the same time that the baby is born and they grow up together. And again, things like that. But for many, especially Americans, typically it is just thrown away as medical waste. However, it can, especially the umbilical cord blood, is something that can be saved and stored. It has some of those omnipotent stem cells that can be useful both for the baby when it gets older. If it needs a little liver, by then we may be able to grow one. Or many people will donate the umbilical cord blood to science for research purposes. So there's all sorts of things that can happen with the umbilical cord and the placenta after birth takes place. All right. Excellent. All right. So we know how progesterone levels need to be high to be able to maintain the uterine wall. We know that human chorionic gonadotropin maintains the corpus luteum early on. But then again, as we talked about, starting around weeks 8, 9, 10, placenta forms, and the placenta is responsible for producing the hormone levels. So human chorionic gonadotropin levels start to drop. This, as you can say, steadily increases over time as the baby is continuing to grow. After all, the baby gets bigger, the placenta gets bigger, and these hormone levels go up. So again, our ovum producing from the trophoblast cells, producing that human chorionic gonadotropin, which stays high for about the first 4 months. Our corpus luteum and then the placenta produces estrogen and progesterone levels. But then the placenta, starting around week 10 through 12, starts producing the estrogen and progesterones instead. But the placenta also starts to produce some other hormones. One of the hormones that it produces is a hormone called relaxin. Relaxin helps to, as the name would indicate, loosen and relax the ligaments of the female to allow for the accommodation of that growth of the baby inside the abdominal pelvic cavity. So again, it loosens the omentums, it loosens those ligaments that we talked about, holding everything in place so that the baby can expand to grow within that place. The other thing that relaxin does is it loosens the fibrocartilage of the pubic symphysis, making the pubic symphysis more flexible to give more give so that baby can be passed through the true pelvis, right? However, remember our pelvis is also our stable base. So as a result of that, that loss of stability gives pregnant, especially late-term pregnant women, a very distinctive gait to the way that they walk. And I still will not say waddle. No, relaxin is not what is responsible for relaxing the cervix. Excellent, because we wouldn't want that happening so early on in the process. The effacing and dilation of the cervix occurs by a separate process. Now, starting near the end of the term, starting at about week 32, we start to see an increase in the amount of human chorionic somatomammatropin that is being produced. Again, a big alphabet soup term that we need to learn, but again, a lot is in the name. Human chorionic somatomammatropin, also known as human placental lactogen, obviously with those two names, involves the maturation of the mammary glands. Males and females that have never been pregnant both have mammary glands. But in females that have never been pregnant and in males, those mammary glands are immature. They are not capable of producing milk. However, starting around month 3, when the placenta forms, the placenta starts producing a hormone called human chorionic somatomammatropin. It continues to build up, and like I said, it peaks at around week 32. And the job of this hormone is actually to mature the mammary glands so that they are capable of producing milk. This typically leads to a swelling of the breasts as a result of that. Prior to that, and again, males and females that have never been pregnant, the breast is primarily adipose tissue. But we start to get the development of those mammary glands so that they can actually become active and produce milk. Now, of course, they don't produce the milk during pregnancy, but they start to mature so that they are capable of producing milk. The glands grow and expand dramatically as a result of that. All right. And we'll talk about that process as well. And also, we see an increase in corticotropin releasing hormones as well. Again, corticotropin, these are the hormones of the adrenal gland, again, playing a huge role in metabolizing, releasing glucose and mobilizing glucose to use energy, electrolyte levels, things along those lines. Because again, remember, mom's metabolism is dramatically enhanced by the pregnancy process. Being pregnant turns women into basically little baby ovens. There have been instances where a male is capable of lactating. Typically, there are 2 factors that can cause it. One is tumor that actually forms. As much as 10% of breast cancers do occur in males. And so it is possible to be a male that has breast cancer. And if the breast cancer occurs in the mammary gland, that actually can cause the mammary glands to abnormally develop and can produce milk as a result of that. Where you also saw cases of this is, again, very early on in the abuse of steroids. When steroids, before we had a better understanding of how steroids functioned, right, when males first started taking human growth hormones and things along those lines and androgens to increase their muscle mass. And then, so again, men started abusing hormones to get bigger. So, bodybuilding companies or, corporations and other things like that, sports started testing for those androgens, for those human growth hormones. And so what they found next is that taking feminizing hormones like estrogens and things along those lines could mask some of those elevated androgen levels. In fact, many of the more famous examples, Manny Ramirez is probably the best example. Manny Ramirez actually got popped from having too high estrogen levels, because that's an indication of that they're using that as a masking agent. So the abuse of these feminizing hormones in some instances led to males then developing breasts and actually being able to produce milk as a result of that. So one of those stereotypical things when you think of those big, huge roid-raged meatheads is enlarged breasts and actually the formation of milk as a result of those masking feminizing hormones. All right, but let's talk about how this changed when it comes time for childbirth. Again, Childbirth, the process of parturition, or what we think of as labor and delivery, is a series of events that expels the infant from the uterus. Labor is typically initiated by hormone changes. As you saw before on that previous map, maybe we should go back to the previous map. Notice, right, starting about 7, 8 months in, we start to see an increase in estrogen levels. And especially near the end, estrogen levels really start to peak as progesterone levels start to taper off. That decrease in progesterone levels and increase in estrogen levels is one of the big triggers that initiates parturition. That corticotropin releasing hormones not only helps to metabolize and increase the metabolism and the resources available for the mom while she's developing baby, but those levels start to rise as well. And these trigger our uterine contraction. As we know, from all the way back in 430, this starts a positive reflex arc, right, where we have that positive feedback, where it starts with baby head down, face forward, stretches, pushes against the cervix. That cervix has stretch receptors, and those stretch receptors fire action potentials. Those action potentials go to the hypothalamus. The hypothalamus produces releasing hormones. No, I'm sorry. The hypothalamus fires an action potential, and neurally triggers the posterior pituitary to release oxytocin. Oxytocin causes the uterus to contract. And when the uterus contracts, baby pushes harder on the cervix. And as it pushes harder on the cervix, we get more action potentials, more oxytocin is released, stronger contractions, and we get that positive feedback reflex arc. The placenta also releases prostaglandins. Remember, we talked about how prostaglandins also can stimulate that uterus to contract. And again, all of these hormones cause a stronger contraction, and this continues to build. This parturition or labor occurs in 3 main phases. The first, as I mentioned, is the dilation. The cervix becomes dilated to a massive, I think it's about like this big, 10 centimeters, isn't that 10 centimeters? Super easy to pass a baby through that. Dilates and effaces, becomes flimsier, becomes looser. And our uterine contractions begin to start, an increase in intensity, increasing in speed. Again, baby typically is head down, face down, for the passage to pass through more easily. If the baby is not face down, head down, then it is typically what we call in a breech position. It is possible to pass a baby in a breech position. However, the problem is the baby is not oriented in a way where it comes out as smoothly. So what happens is if you have to try to grab a limb and pull it up by the limb or something like that, it's more likely something would get stuck. It's more likely something would get damaged. It can be more likely that tearing or injury can occur to mom inside. So again, it is a more difficult process with more inherent risks. Can it occur and occur safely? Yes. But especially since most delivery takes place in HMOs and they're not willing to take the liability risks. If a baby is in a breech position, then typically what they will do is they will take the mom to a surgical suite. There, they may try to manually, both internally and externally, manipulate the baby to rotate it into the proper position. But that type of manipulation can increase the contractions, which can decrease blood flow to the baby. And so if baby's heart rate gets too high, if baby's oxygen levels get too low, if there's too much stress on mom, then the reason they do it in that surgical suite is they will instantly do a cesarean instead of that. So really, the main reason that we don't do it is that it is a more challenging, more risky for baby and for mom, just harder to get the kids out that way. And the insurance companies don't see the added risk as worth it, right? Especially with what's the term I want? The modernization is not the right word that I'm looking for, but the process of a cesarean section has been dramatically improved since it first began. Typically, when a cesarean section was first identified and discovered, again, they basically just split you down the middle and pulled it out. They typically now are much more precise in where the incision's made. When at first you had a cesarean, basically that meant all additional childbirths had to be cesarean as well. That is no longer the case. Now, if you give a cesarean birth, you are able to still have a normal childbirth, non-cesarean childbirth for subsequent children after that. So the risks associated with a cesarean section are so much lower than those of passing a breech baby that's why they do it that way. All right. The other thing that forms is an amniotic sac. That amniotic sac is part of the development from that inner cell mass, and it is filled with amniotic fluid. Anybody know what amniotic fluid is? Nutritious water? It is definitely water with nutrients in it. Where does it come from? I like that, nutrient water. Where does that nutrient water come from? Where does the amniotic fluid actually come from? Interstitial fluid's not a bad guess, but nope, it's not interstitial fluid.
That's where the nutrient exchanges occur between babies and mother.
It's not from the exchange of mom. It's not from the placenta. Where it actually, there we go. Where it actually comes from is from the baby's kidneys. Exactly. Amniotic fluid is baby pee-pee. Obviously, baby's lungs aren't working. It's not getting any air. Baby's digestive system isn't working. It's not getting any food. But does baby need to be filtering its blood? Absolutely. And so the amniotic fluid is actually the urine from the baby. It's one of the reasons they monitor amniotic fluid levels so closely. If your baby doesn't have enough, if there's not enough amniotic fluid inside of the uterus, that could be an indication that the baby's kidneys are not working, are not functioning properly. So they will typically do an ultrasound to take a look to make sure that there isn't a blockage of the uterine tubes, the blockage of the ureters or anything along those lines for that process. That's an interesting way of looking at it, Alex. Absolutely. I don't know how much swallowing actually takes place. There's not a lot of digestive function that's going on with the baby in that sense. So I wouldn't say that it's necessarily recycled because the digestive system really isn't that functional at that point. And again, it's not breathing at that point, so anything along those lines. So I don't think that that's so much of not that it would be a concern anyway. But yeah, basically amniotic fluid is actually the baby's urine. All right. Once fully effaced, once fully dilated, then powerful uterine contractions lead to expulsion. Baby is passed through the cervix and through the vaginal canal. Good question. I'm not familiar enough with amniotic dermatitis to know the cause of that condition. So I mean, that in the uterine wall are the only thing that the baby is in contact with in utero. So possibly, but I'm afraid I'm not familiar enough with that condition to be able to say. What does it mean when someone's mom's carrying the baby in the lower back? Well, obviously you don't carry it in the lower back. My guess is that it has to do more with the position of the baby in the uterus. Sometimes they sit a little higher. When they tend to sit a little higher, you typically have more respiratory issues that way, where again, because it's so close to the diaphragm, it's hard for the diaphragm to move down. So breathing can be a little bit more labored. Whereas when the baby's a little bit lower, it tends to sit more on the bladder. I notice as we look at this picture here, here is the bladder. Here you can see how it's being squeezed. So again, you get these moms who are pregnant who have to go running to the bathroom because they feel like they're going to explode in urine if they don't get to the bathroom, and they get there and just the tiniest little tinkle comes out because, again, of that pressure on that. So my guess is that maybe a lower position might put more stress on the lower back that way. So that would be my guess what they mean by that, by carrying the baby in the lower back. It's just probably the baby's probably more descended, and then it affects, because again, remember, this is going to affect your balance and equilibrium. It could put more stress on the lower back. So my guess is that's what they mean by that.
I have a question.
Yes.
Is there a cork that blocks the cervix before expulsion, so it needs to be removed?
Great question. Yeah, actually, this is something we didn't talk about before. So now that you mentioned it, I'm glad you mentioned this because I realized we didn't talk about this before. There is, remember, as we talked about, the female reproductive system is open to the outside world. And as such, there is actually a structure called a mucus plug that actually fills the cervical canal. This mucus plug is a thick, viscous mucus plug that, again, helps to stop movement of materials from the vaginal canal into the uterus and to the rest of the body. The hormones that are released, luteinizing hormone, that spike of luteinizing hormone, not only does it trigger ovulation, but it also thins that mucus plug, making it more watery, making it actually easier for the sperm to get through it, so that fertilization is more likely to occur. Of course, then once luteinizing hormone goes away, that mucus plug thickens again and stays there until dilation starts. At the start of dilation, typically that is when the mucus plug is released, and also when the amnion ruptures. If the amnion doesn't rupture, what they'll actually do is sometimes, and again, if expulsion is delayed, they'll use a hook and go in there and tear it, and then that releases that fluid, and often that can trigger the uterine contractions that can cause expulsion. One of the concerns, if the water breaks prematurely, is that when that water breaks and flushes that plug out, then basically baby is exposed to the outside world and all the harmful pathogens that are out there. So typically, if expulsion doesn't occur within 24 hours of a woman releasing her water or having her water break, then they will typically either try to chemically induce labor, or they will ultimately do a cesarean.
I want to ask also, so that is why, because I had this story when I was pregnant with my second son. I was having I was carrying heavy stuff. That was, I told why it happened. So, and this plug was removed, I think 2 weeks before my expected labor.
Okay.
And the doctor said that they need to force the labor because now we can't wait until because of this plug.
Right, exactly. Because now, again, you run an increased risk of either you or the baby coming in contact with more harmful pathogens. As again, we know bacteria love warm, moist nutrient-rich environments. And so again, it can dramatically increase the danger for the baby and for the mom. And so yes, they're more likely to want to induce labor. And again, if you're only 2 weeks away, then again, at that point, the baby is pretty much fully baked, anywhere between 36 and 40 weeks. So that shouldn't have been a major concern that way.
Yeah, it was not. Everything was
Yeah.
good.
Awesome, excellent. Yeah, everything is kind of moved around and pushed up towards. In fact, I haven't looked at it, but I have to imagine it's there. If we look at the chart, here we go. So notice here, we have the very beginning of the process, our little developing embryo in the uterus. Right here we see the bladder, here we see the ovary and the uterine tubes and all of those things. And again, remember, this is going to continue to expand to become this. All right, there's the bladder shoved up against the pubis. Right, here's the head down towards the cervix. Right, the digestive system, the rectum and the sigmoid, and everything is pushed up. There's the placenta. So yeah, obviously, as you can see, everything is kind of shoved out of the way as that baby is growing and developing inside the abdominal pelvic cavity, pushing all those organs up towards the diaphragm and more. When the water breaks, typically you're just releasing fluid out of your vaginal canal. So my understanding is that it doesn't feel like urination. It's not going through the urethra, but suddenly your legs and the floor beneath you are covered with water. So I don't think, it's definitely not a painful sensation when it occurs, but again, yeah, you're aware of it. There you go, a lot of pressure coming down. Okay. All right, so powerful, painful contractions, right? The passing of a basketball through your pubic bone. All of these things are the pubic, the pelvis. And then once baby's out and it cries and everybody's all excited and paying attention to that, but the process is not completed. Nobody's usually paying attention to the last part, but we still need to expulse the placenta as well. While everybody, the nurses, mom and dad are all paying attention to the baby, the doctor needs to make sure that the placenta is fully released from the uterus. They inspect the placenta to make sure that it is complete and intact and that there aren't any pieces left inside. And if there's any concern, they will actually go in and scrape the functional layer of the uterus to make sure that all parts of it have been removed. And again, often, nobody's aware of this even happening because the expulsion of the placenta is a fairly uneventful event in most instances. And again, everybody's focused on the baby. And so it's not, and obviously it doesn't have the structure, the mass that the baby does. So it typically passes without any issues at all. All right, again, dilation can take anywhere from hours to, in some cases, days, 36, 48 hours even to fully dilate. Expulsion can take anywhere from 30 minutes to several hours, 4, 5, 6 hours at the most. And then this placental stage usually takes 30 minutes, if even that. All right, questions on that? All right, the last little bit of anatomy we have to talk about. Once baby's out, we need to feed it. And there are, of course, two, both equally acceptable, viable ways of doing that. You can use formula, because again, our formula has definitely gotten very, very sophisticated, but there's also a breastfeeding. Great question. So, yeah, you will get additional bleeding, especially if they end up scraping the wall. The functional layer can be irritated for a while. And again, it takes a while for all the hormone levels to drop, and so bleeding can last. 6 weeks seems slightly on the higher end of that, but it can be less than that. But 6 weeks is definitely a reasonable amount of time for that. Also, as we'll talk about, and one of the advantage of breastfeeding is it helps the female body to recover. Part of that recovery is decreasing the size of the uterus. Now, the uterus, remember, started something the size and shape of a pear. It's never going to get back down to that size again, but it will get significantly smaller again. However, with a larger uterus, you have a larger functional layer. And so it is not uncommon for the menses of a female after pregnancy to be larger and longer than the menses was prior to the pregnancy. All right, here we see the anatomy of the breast. Again, as we talked about, it is primarily adipose tissue. But we can see also within here the formation of some mature mammary glands, big lobular glands segmented out. Notice the adipose is sustained and anchored in place, keeping the breast in an upright position by these suspensory ligaments, attaching it to the muscle like the pectoralis major. And of course, we know the function of these mammary glands is to produce milk for the baby. As you can see, there are multiple lobes to these glands that then feed out into a lactiferous duct into a big full space called the lactiferous sinus. And then it's the lactiferous sinus that's opening is within the nipple. You can see that there are multiple openings because there are multiple lactiferous sinuses. All right. Notice also there is a darker pigmented region around the nipple known as the areola, and that areola actually becomes typically darker pigmented upon pregnancy. And why might that be the case? Exactly. Babies don't see so good. So the increased contrast can actually make it easier for the baby to be able to find the nipple and to latch onto it. That is, as we talked about in 430, an inherent reflex. When a baby is born and you touch them on the cheek, they will rotate towards that and try to latch on. And so it's a reflex that they are born with. Now, milk production is stimulated by what hormone? What hormone causes milk to be produced? Prolactin, absolutely. Well, so again, let's be careful. You're absolutely correct. Milk production is controlled by prolactin. All right. So that causes the lobes to produce and swell with milk. But you guys are correct in that the letdown of that milk, and by letdown, what we're really meaning by this is the release of the milk from the lobules into the sinuses so that it can be more easily expressed. And that letdown, you are absolutely correct, is stimulated by oxytocin. So there are indeed two hormones involved in the production and release of the milk, but production of the milk is produced by prolactin. And again, what hormone causes these to mature and get ready? Is no one just willing to say it? Is that what it is? There you go. Human chorionic somatomammatropin. Exactly, Ashley. We will talk about that in a minute, but you've got the right idea. Oxytocin causes the milk to be let down, but it also causes uterine contractions that helps the uterus get back down to its original shape. So breastfeeding is both beneficial for mom and baby physiologically. right? And that doesn't even include the emotional attachment that forms from that skin to skin contact as well. That bonding. Remember we talked about in males how oxytocin is that cuddle hormone produced from intimate contact. That skin to skin increases oxytocin levels in both mom and baby and helps in that bonding. So you can get an enlargement and swelling and congestion of these ducts, congestion of these sinuses can occur during this. Can be irritation from the expressing of it. Bacteria from baby's mouth getting into the openings can cause problems. There are a couple things that can cause challenges that way. So again, we have the pectoral fat pad we talked about. We talked about the nipple, areola, the suspensory ligaments. And I don't know if you guys are just a completely quieter class or what, but one of the questions I usually get about this time is, again, we were talking about those suspensory ligaments, and the goal is to maintain the breast and keep that breast in a more upright position. But as we know, as we age, those suspensory ligaments can start to stretch. And so as a result of that, the breasts can start to descend. Right? So usually the question I get around this point is, so then what's better? Is it better to wear a bra or to not wear a bra? So since you guys didn't ask me, I will ask you. Is it better to wear a bra so that you're not putting stress on those suspensory ligaments and it maintains the suspensory ligaments longer? Or is it better to not wear a bra so that you build up strength in those suspensory ligaments and it helps to maintain them in place? All right, Alex is not wearing one. I've got 2 wear, I've got 2 not wear, a lot of free the boobies. More better to be aware. I'm not sure you're allowed to have a vote, Alex. So, believe it or not, there have actually been studies that have looked at this, and what they found is that it's basically inconclusive. You do have people on both sides that feel very strongly on both sides. Some people feel very strongly that you need to build up the strength in those suspensory ligaments, so it is important not to wear a bra. There are others that truly believe that you have to wear the bra to maintain those suspensory ligaments so they don't stress out. And so far, there hasn't a lot of research that's been done on it, but the research has been inconclusive. So it doesn't matter. Wear your bra, don't wear your bra. No rules. It's not going to affect the attentiveness of your breasts one way or another. Yeah, there you go. Wear one half and not the other. There you go. It's the easiest way to do it. Do your own scientific experiment. All right. Exactly. It's a ligament. So, but again, the more collagen fibers you have in it could cause it to strengthen. No, because again, remember, it's not a muscle. This is like, as Levon pointed out, it is a collagenous ligament, right? So it's a collagen fiber holding it in place. Doing exercises isn't going to help to maintain the strength of that ligament. All right. Well, okay, true. You can have surgeries to either basically draw the skin up, which is what they do that to help to maintain it. If you don't like the size, there are implants that can be placed, typically either behind the fat pad or in some cases underneath the muscle to increase the size of it that way. They used to make an incision under the breast and they would insert it that way, but you would then have scarring under the breast. So now what they actually do now is they actually go through your belly button. They go through your belly button up to the breasts and put in an inflatable device, which they typically fill with saline to the level that you require to, so you can get your mommy makeover that way. All right. If you remember back into 430, these do indeed, they are a modified sudoriferous gland. I have not heard of adipose implants. Again, not my area of expertise, so it wouldn't surprise me. But saline, my concern about adipose is that, I mean, unless they're taking it from another part of your body, you might run the risk of, if the wall of the implant fails, then some generic adipose could cause an autoimmune reaction of some type. Saline tends to be fairly, they used to use silicone because silicone had a more natural feel to it. But again, they had a lot of issues with leakage and intolerances that formed because of that. You get the best of both worlds, right? You get rid of your love handles and put them in your boobies instead. Sure. Again, the things that people do for vanity no longer surprise me. So I have not heard of that, but I wouldn't be surprised by that at all. All right. Back in 430, we talked about how these are a modified pseudoriferous gland. So they're primarily tubular in shape. But if you remember, way back in 430, we talked about how there are 3 modes of secretion. Merocrine, which basically is exocytosis, holocrine, which is where the cell accumulates and ruptures, and epocrine, where the cell swells, the top pinches off and releases. We talked about how anal glands of dogs use that apocrine mode of secretion, and there weren't any confirmed cells that do. However, what is being studied are these mammary glands, because these mammary glands basically produce 2 types of milk. There is a fore milk and there is a hind milk. The fore milk is more watery, more glycogen rich. The hind milk is thicker, more viscous, and has much more fats and protein in it. So it's believed that it is possible that with the 2 different types of milk, there might be 2 different types of secretory structures, and the hind milk may actually be released via an epocrine mode of secretion. But that isn't something that has been confirmed. These 2 different types of milk that are produced by the breast are one of the reasons why when best breastfeeding, it's usually best to let the baby completely drain one breast and then switch to the second. And the next time start on the second breast and make sure you drain it completely. What can happen with some babies, and what happens is the fore milk is much easier for the baby to get. So they will drain off the fore milk and then they'll get fussy. So mom switches and they drain off the fore milk and they're not completely draining the breast. The issue with this is that the baby is getting that more watery, more glycogen rich milk. And as you know, when you eat a very carbohydrate rich meal, you're hungry again an hour later. So typically the babies feed much more quickly because they're not being fully satisfied. Whereas if you can drain it and get that protein and get that lipid into the baby, then it is going to be sated for a longer period of time. One of the beauties of this process also, and we'll talk about the hormone regulation of it in just a minute, is it is completely need-based. Now, again, I'm talking in generalizations. Are there people who have difficulty with breastfeeding or unable to breastfeed and things along those lines? Yes, those types of things can occur. Some of the early chest implants, typically when they were put to the front of the breast, that would disrupt the glandular activity. And that's the other reason why they go behind it or even in the muscle now. You can actually have breast implants and still be able to breastfeed. But there are some people who have difficulty or problems with it. And again, this is nothing to be embarrassed about. It is common occurrence. And again, formulas are a perfectly acceptable way of feeding the baby and providing the oxygen and providing the nutrients that that baby needs. Although there are some benefits to breastfeeding, like we talked about the bonding that comes from the skin-to-skin contact that comes from that, the benefits to mom of the oxytocin being released to cause the uterine contractions to help her to recover. And remember, as we also talked about earlier in this semester, the antibodies. Mom is providing that naturally occurring passive immunity by passing her type A antibodies onto baby from the milk. So there are definitely some benefits to breastfeeding that you don't get from formula. But if you're unable or unwilling to breastfeed, formula is a perfectly acceptable alternative. All right, mammary glands fully developed by month 6. Again, we talked about the 2 hormones that are responsible. However, when baby first comes out, mom's typically not producing milk right away. Right? Baby's not too hungry after the move anyway, but for the first couple of days, they're producing a liquid gold known as colostrum. Colostrum is a very, very protein-rich, very low-fat dense material that is, or substance fluid that is being produced. However, in time, mom's milk comes in, and the more you breastfeed, the more milk you're going to produce. So again, typically if you have twins, as long as you continue to breastfeed, you're going to be able to continue to produce milk. The longer you breastfeed, the more milk you're going to produce, right? And we've known about this and taken advantage of this for generations. Back in medieval times, right, the queen was too busy chopping off people's heads to have time to breastfeed her own baby, so they would get a wet nurse. What good is a wet nurse? Who is a wet nurse? What is a wet nurse? Yeah, a wet nurse would be some peasant woman who had just had a baby at the same time that the queen did. The queen would move her into the castle, and that wet nurse would feed both her kid and the queen's kid. So exactly, it was somebody else who was able to produce milk. And again, so some of these wet nurses were highly coveted. When the queen was done with her, maybe she would gift the wet nurse to some duke's wife who had just given birth as well. And again, is it possible for a woman to produce breast milk for 2, 3, 4, 5 years? Yeah, absolutely. And again, not just in these wet nurses who were doing it for these purposes, but some women will continue to breastfeed their kids till they're 5, 6, 7 years of age. Now, again, hopefully by the time they're 5 or 6, it's not their primary source of fuel and energy and food, right? Again, if the kid can ask for it, then it's kind of awkward to be doing that. But they often will do it as a bonding thing or as a nighttime ritual for going to bed or things along those lines. When I was in graduate school, there was a woman in my cohort who breastfed her kids until they were basically 8 years of age. So again, third graders. So again, to each their own how they do that. Again, but it is quite possible, quite capable to continue to produce milk for a long period of time. And again, is everybody going to be able to do that? No. But in an ideal situation, it is use dependent. The more you use it, the more you're going to be able to produce.
Why some moms say that they can't feed like more than 2 months?
So again, I'm painting with broad strokes here. As I mentioned, there's some people who aren't able to do it for real life issues. They have to go back to work. They may try pumping and things along those lines, but typically pumping isn't as effective at draining the breast the same way. And if you're not able to drain the breast the same way, you're not going to be able to keep that hormone cycle going to maintain it. So that can cause it. Some people's babies are more fussy. Nipple size can be an issue. Absolutely. There's lots of factors. As I talked about, the baby's got to do some of the work of getting that hind milk out of the breast. And if the baby's impatient and not willing to work to get that hind milk out, that can cause difficulties. And again, rather than working through them, since there are viable options, there are other alternatives to things along those lines as well. So there are many factors that can influence and affect it, both biological and societal.
They say they don't have it. Like they say it ended in 2 months. Is it possible that it can end in 2 months?
Yes, it is absolutely possible that could happen as well, that it can dry up. While it is primarily based on production, other hormones, as the woman's cycle changes and goes back, as if her cycle resets too quickly, that can cause a decrease in milk production and things along those lines. So sure, absolutely. There are biological reasons why milk production can decrease. Absolutely.
Okay, thank you.
Yep. I think we kind of already hit on this, but you got the idea in that when baby suckles on the breast, stimulates the nipple, that stimulation of the nipple sends a neural signal to the hypothalamus, which triggers the posterior pituitary to release oxytocin, and that causes the oxytocin, the milk to be let down, the ejection of the milk. All right. That also stimulates prolactin to produce the milk as well. I make a point of mentioning this because typically you breastfeed babies when they cry. So your baby cries, you grab the baby, you feed the baby, and again, the stimulation of the nipple causes the milk to be ejected, to be let down. But then after a couple months, you go out, again, grandma finally comes over, you get a night out with your husband, and again, a nice fancy night out, which means going and walking around the mall. And as you're walking around the mall, someone's other kid cries. And what happens when that other kid cries? Yeah, exactly. That the crying of someone else's baby can actually cause the milk to let down. The other way we take advantage of this is, as I mentioned, for pumping. Many women have to go back to work before they're done breastfeeding, and so they can pump because, again, you should be emptying the breasts every 3 hours or so. So what they will do is they will have videos that they can watch of their baby, or they'll have audio recordings of their baby where they see the baby, they hear the crying, and those stimuli can help to trigger the letdown mechanism to make pumping more efficient. All right. Questions on that? Well, with that, that is everything you are responsible for in anatomy and physiology. We are done. All right, like I said, it has been an amazing semester. It's been an unusual semester, a unique semester, but like I said, it both feels like it's gone super fast and it's been going on forever. So I want to thank you for your time and diligence. Again, I will still have my normal office hours over the next week while we have our exams. So if you have any questions, email me or come to my office hours. Read the questions carefully on the exam so that you can answer them correctly. As I've said many, many times, people often lose points not because they don't know the information, but because they don't read the questions carefully. Read the questions carefully so you can answer them correctly. I wish you all tremendous success moving forward. Good luck to you in your chosen careers as you move on from this and the rest of your academic careers. I wish you all great success. It has been like I said, it's been a unique semester. So thank you guys so much. I appreciate it. And take care.
Thank you, Professor, very much.
All right, guys. Have a good day, and I will see you. Good luck on the exam.